What are antibiotic mechanisms of action for med students? If you are a medical student curious about how antibiotics work, you are in the right place. Understanding antibiotic mechanisms is key in medical microbiology, especially during your first and second years. Let's break down the main ways antibiotics take action against bacteria. First up is the inhibition of cell wall synthesis. Bacteria have a tough outer layer called the cell wall, made of peptidoglycan. Antibiotics like penicillins and cephalosporins target specific enzymes that help build this wall. When these antibiotics interfere, the wall becomes weak. This leads to the bacteria bursting due to pressure from inside. This method is effective against bacteria that are actively growing. Next, we have the disruption of cell membrane integrity. Some antibiotics, such as polymyxins, attach to the bacterial cell membrane. This action increases the membrane's permeability, causing essential substances to leak out. When this happens, the bacteria cannot survive, leading to cell death. This method is particularly useful against certain gram-negative bacteria. Another important mechanism is the inhibition of protein synthesis. Bacteria need proteins to function, and they use ribosomes to make them. Antibiotics can bind to different parts of the ribosome, blocking the process of making proteins. For example, aminoglycosides cause errors during protein production, while tetracyclines prevent the attachment of transfer RNA. Depending on the antibiotic and its concentration, this action can either stop bacterial growth or kill the bacteria outright. Then, we have the inhibition of nucleic acid synthesis. Some antibiotics target the enzymes that help bacteria replicate their DNA or make RNA. Quinolones, for instance, block enzymes that are crucial for DNA unwinding. This leads to DNA damage and ultimately bacterial death. Rifampicin works by stopping RNA synthesis, which is also lethal for bacteria. Lastly, antibiotics can inhibit metabolic pathways, particularly folate synthesis. Bacteria make folic acid, which is essential for producing DNA and RNA, differently than humans do. Sulfonamides mimic a substance called para-aminobenzoic acid, blocking a key enzyme in folate production. This action reduces the bacteria's ability to grow and multiply. For medical students, knowing these mechanisms is vital. It helps you understand which antibiotics to use for different infections and how to anticipate resistance. Combining antibiotics that target different mechanisms can improve treatment effectiveness and reduce the chance of resistance developing. Understanding whether an antibiotic is bactericidal or bacteriostatic can also guide your decisions, especially in severe infections. In summary, grasping how antibiotics work lays the groundwork for your studies in medical microbiology. This knowledge will be crucial as you move forward in your medical education and clinical practice.